The eutrophication of Lake Erie has once again resurfaced and is a serious concern for the countless species in the environment and anyone who depends on it for clean drinking water and habitat. Eutrophication promotes deadly algae blooms, an unnatural phenomenon which threatens the biodiversity and health of the lake. Algal blooms are disruptive to the ecosystem for many reasons, such as decreased oxygen content within the water, preventing sunlight from reaching marine life at greater depths, such as seaweed, preventing oxygen exchange from the surface of the water, and does not allow the lake to be used as a clean source of drinking water. Additionally, the more recent introduction of invasive species, like zebra mussels, has contributed to the return of algal blooms in the lake. Within this report, the cause of algal blooms will be explored, as well as a potential solution, implementations, measures of success, and the biological ramifications of our solution.
Lake Erie is once again experiencing another life-threatening eutrophication event. Eutrophication occurs when lakes gather excess levels of nitrogen and phosphorus, along with other nutrients. This leads to toxic algal blooms. Algal blooms can become deadly because when algae grow in excess, they block sunlight from reaching the naturally occurring flora at the bottom of the lake. This prevents other plants from photosynthesizing and producing oxygen, which reduces oxygen levels and the general plant population. To worsen matters, certain types of microbes that feed on decomposing algae rapidly multiply and create harmful bacterial blooms in conjunction with the algal bloom. These bacteria use up leftover oxygen to break down the algae. In mass numbers, they can lower oxygen levels in the water to so little that the water becomes anoxic to fish and other lake-dwelling organisms. This kills off huge populations and damages the ecosystem. But where do these extra nutrients come from? Nutrient excess is claimed to originate from massive increases in nitrogen additions to the biosphere through the production of fertilizers and increases in fossil fuel emissions. Some sources include runoff from farms, which is filled with nutrients from manure, fertilizers, feed, and other farm waste, sewage and industrial effluents, for example detergents, and urban development.
Phosphorus pollution and runoff from outside the lake contributes heavily to the cause of dangerous algal blooms, but factors inside the lake help speed up these blooms and make them even larger. In 1989, an invasive species of mussels, zebra mussels, were found in Duluth Harbor, Lake Superior. Since then, these mussels have proliferated to all of the Great Lakes and are especially abundant in Lake Erie. These mussels help contribute to the algal blooms by eating the competitors of the harmful algae while rejecting the blue-green algae themselves, thus making it significantly easier for the algae to grow by giving them less competition for nutrients. This has been shown to even cause algal blooms in lakes where phosphorus intake is low because of how much they affect the competition.
A riparian buffer is a collection of ordered zones of vegetation planted on the land surrounding the lake, so runoff is forced through a biological filtration system to excrete nutrients before the water enters the lake. In our solution, the riparian buffer will be designed so that zones will progressively absorb more nutrients depending on the type of vegetation we use. The diagram on the right explains this. A study was done by the American Society of Agronomy in 2015 to test if the size and type of vegetation would have significant impacts on nutrient absorption, focusing on phosphorus and nitrogen. Results showed that riparian buffers that had a width of 60 meters, composed of woody soil, would retain 99% of phosphorus and nitrogen after every heavy rainfall, preventing nutrients from passing through as runoff. Along with buffer zones around the lake, creating additional riparian buffers around farms and nearby streams would act as primary defense systems against runoff accumulating in rivers. These bodies would otherwise become concentrated in nutrients, later polluting the lake. The buffer zone will be about 60 meters wide and composed of hardwood trees to maximize nutrient absorption, as mentioned in the previous slide. There will be areas around the lake where creating buffer zones would not be possible due to already existing settlements like houses and other buildings. Having additional buffer zones around high nutrient pollutant areas such as farms would further ensure that almost no nutrients are entering the lake in the form of runoff.
Since this problem is not one-dimensional, a solution that attacks all causes for these algal blooms is necessary, and so, not only do buffers affecting phosphorus intake need to be put in place, but a solution to the dangerous invasive species must be found to fully fix this issue. Proliferation of native species that can naturally prey on these mussels could solve this, as it would lead to an increase of natural algae competition, reducing the amount of nutrients going to the harmful blue-green algae. A perfect candidate for this job is the lake sturgeon, a native species in Lake Erie that is currently endangered. This species has been shown to eat zebra mussels in lakes they both inhabit, such as the St. Clair River. An increase in sturgeon would both reduce mussel population in Lake Erie, making algal blooms harder to produce in low phosphorus conditions, while also increasing the population of a currently endangered species.
Producing a riparian buffer requires money, time, land, research, and workers. During the implementation process, careful consideration must go into choosing the right plants, placing such plants, and how they will be maintained. When selecting species for a riparian buffer, they should be collected based on their ability to absorb a large nutrient load, but also for their ability to acclimate to wetter environments and their resistance to harsh weathering. For example, semi-aquatic plants such as bulrushes should be planted closer to the water's edge where trees are unlikely to survive. But land surrounding farms that is further from the shoreline should consist of hardwood trees such as oak for their capability to remove extreme amounts of phosphorus from runoff. Each plant should be placed roughly 2.5 meters apart to ensure effective absorption. About 574,000 bodies will need to be planted. Given that each tree will cost roughly 10 cents given the cheapest means possible, the riparian buffer system should cost about $57,400.
The implementation of proliferation of lake sturgeon populations in Lake Erie would likely be done through transplanting of eggs from healthy populations of lake sturgeons, such as St. Clair River, raising them in a safe environment, and then re-releasing them in Lake Erie. This would be relatively cheap, but would require many people as you'd have to harvest the eggs, find a place to raise them, and then re-release them after you've tagged them all. The biggest difficulty of this solution would be finding a place to raise these eggs before you release them, likely a zoo or some other animal institute.
You may be wondering why this solution is superior. The utilization of both the riparian buffers and natural predators of zebra mussels offers a two-pronged approach. The solutions proposed offers a sustainable yet long-term solution. The buffers planted will continue to grow for a long time, and the absorption of nitrogen and phosphorus will increase with age, with the added benefit of absorbing carbon dioxide and producing oxygen. Additionally, the plants used as buffers will act as anchors to prevent soil erosion. The implementation of the buffer also has great potential to aid in the conservation of Lake Erie by preventing lakeshore development. Lake sturgeon will also be given a chance to increase in abundance, which will help to restore their endangered population. As the lake sturgeon grow in population, they will require more zebra mussels for sustenance. This will, over time, reduce the invasive zebra mussel population. The solutions mentioned within this presentation are generally cheaper and better for the environment than alternative solutions, such as using a bubbler. Bubblers prevent algal bloom, but requires industrial-level construction in order to keep the water remaining stagnant, which in turn prevents algal bloom from occurring.
In order to measure the success of the riparian buffer, measuring the phosphorus and nitrogen in both the buffer floor and within the lake will determine how much phosphorus was prevented from entering the lake. Additionally, the oxygen levels within the water should be measured in order to demonstrate the effectiveness of algal bloom reduction. The data collection would be most accurate if computed using a nuclear magnetic resonance spectroscopy, which is used to show a complete breakdown of elemental composition. By comparing samples from before and after implementation, it is possible to identify weak spots and the effectiveness of the plan. Oxygen in the lake should be measured using a dissolved oxygen meter and sensor. This can give a good idea of how oxygen levels are improving over time when measurements are taken consistently after heavy rainfall or weathering activity. The metrics of fish health and hypoxia could be measured through both tagging, through the mark recapture method, and through fish biopsies or autopsies. Of course, visual inspection of the lake could be used to make observations of any improvements or deteriorations of lake health.
This process of introducing a buffer system will no doubt be beneficial to the environment as a whole. Despite this, some ramifications may occur. Planting more trees, for example, will increase competition within the area for pre-existing trees. As a result, the natural balance of nutrients within the soil may be disrupted, which could lead to nutrient depletion within the soil. There is also the potential that riparian buffers will cause a decline in species diversity according to the Shannon Index. This means that the ecosystem would be more susceptible to the spreading of disease, which could lead to biodiversity loss.
While the biological ramifications of an increase in lake sturgeon population in Lake Erie are few, there are a few worth mentioning. It could cause an overabundance of lake sturgeon in the lake, and while they are native to the lake, this rapid increase could be dangerous to the current balance of the ecosystem. Additionally, they can live for up to 150 years and do not stop growing, which means there is potential to reach a point where the sturgeon negatively impact the food chain by overconsumption of prey other than zebra mussels, which could result in reaching the carrying capacity and significantly lowering populations of other animals in the lake.